Thank you very much, Vanessa. Much appreciated, and a very warm welcome to everyone to today's Tea Time webinar. Today's webinar will be presented by one of my colleagues from the European team, Edward Grahame, who is Field Application Scientist based in Glasgow in Scotland. And today he'll be talking about GenVoy-ILM, a tool to rapidly accelerate preclinical vaccine development. Just to introduce some more of us on the Europe team. So I'm Richard, the Regional Director, and work alongside my colleagues. So Jürgen, based in Berlin, and Suha, based in London, are two regional sales managers available for any commercial related inquiries, whereas for scientific applications and technical related inquiries, Edward in Glasgow, Martin in Leipzig, and Sarah in Paris. We'll all be very happy and welcome to hear from you and to field any questions that you might have. So thanks once again, everyone, for attending. We very much appreciate your coming along today. And now it's my pleasure to hand over to today's speaker, Edward Grahame. Thank you.
Thanks very much, Richard, for the introduction. Yeah, it's really nice to be able to present to you today on GenVoy-ILM, on a project that I was lucky to be involved in, and kind of show you how you can use this tool along with Precision NanoSystems' other offerings to really accelerate that kind of early development vaccine stage for RNA-LNPs. So I'll start with PNI's mission statement, which is that we aim to accelerate the creation of transformative medicine that significantly impacts human well-being. And we really do that as a company by being the go-to solution provider for anything lipid nanoparticle related. So we're most famous for our manufacturing technology. So that includes the NanoAssemblr platform, which is based on next-gen microfluidic mixing technology for controllable, reproducible formation of lipid nanoparticles. And then we also have our nanoparticle technology itself. So this is a series of research-only reagents, as well as lipids, or lipid mixtures, should I say, that are available for clinical and commercial licensing into your drug product. And it's that together with our services that as of next year, we'll be able to offer out of our BMC in Vancouver, Canada. So that includes GMP manufacturing, so ways we can directly support you. It's these kind of three main pillars together with our expertise and our patent space that makes PNI unique in terms of lipid nanoparticles. And so the NanoAssemblr platform, as I mentioned, is what we're most famous for. It's our next-gen microfluidic technology implemented into a series of instruments, each designed to handle different volume scales from discovery in the Spark instrument through to the GMP scale, with everything in between, like the Ignite, dealing with formulation development. So this is where you're looking at process parameters combined with formulation elements, and then scale up on the Blaze instrument. And then obviously from there onto clinical. And with our recent acquisition into Danaher, essentially in terms of being able to make LNPs, make RNA-LNPs, we have solutions upstream and downstream now of the kind of particle formation step. So where Precision NanoSystems supports you is in microfluidic mixing and inline dilution. So these kind of critical process steps. But upstream of that, we have mRNA synthesis. So we have capabilities there with our colleagues at PAL and Cytiva. We have ways to be able to offer you solutions to process the particles, diafiltration and concentration through TFF, sterile filtration, and onto fill finish. So we really have that complete end-to-end solution in terms of manufacturing. But manufacturing is only one element in what we would call the genetic medicine toolkit. And certainly in the context of an RNA-LNP vaccine, you need a robust manufacturing platform that will allow you to produce vaccine at scale. But you also need the genetic payload platform itself, and you need a lipid delivery to be able to produce that. And so it's our GenVoy delivery platform and how that kind of interlinks with our manufacturing platform that I want to talk about and touch on today. So the recent pandemic has seen the emergency authorization of 2 RNA-LNP vaccines, which everyone is aware of, the vaccine from Moderna and the vaccine from Pfizer-BioNTech. And really what the kind of development and authorization of these is highlighted is that RNA-LNPs can respond incredibly rapidly in a pandemic situation. They're incredibly advantageous in a vaccine context. And whereas with previous kind of vaccine modalities, it often takes a very long time to be able to develop. RNA-LNPs afford that kind of true platform technology where you can quickly adapt to changes in the disease, so switching out aspects in the RNA design to be able to account for new kind of pandemic changes like new disease strains as they emerge. And then coupled with a kind of robust manufacturing platform, you have a way to make them safely and efficiently at low cost, and something that can scale quite quickly from bench through to clinic. And so that brings me on to talk about GenVoy. So within our GenVoy umbrella, we have a series of lipid reagents, some of which are designated for research use only. So we have a product that's concerned with ex vivo delivery to T cells. And then we have our GenVoy-ILM product, which is what I'm going to talk about today. And this is a research use only reagent, which comprises an ionizable cationic lipid, so the function of which is to bind your nucleic acid during manufacture, and then also help in facilitating endosomal escape. And that together with cholesterol, with a helper lipid, which is DSPC in the case of our GenVoy-ILM product, and a stabilizer, which is there to be able to produce your particles and keep them from essentially agglomerating in suspension. That together provides a lipid mixture that you can take and use on the NanoAssemblr platform to really accelerate that early RNA vaccine development workflow. So if I come on to my next slide. So essentially, GenVoy is to rapidly accelerate RNA development. Really what GenVoy is it's a off-the-shelf, ready-to-use lipid mixture that when combined with the NanoAssemblr platform, so in this study, I was particularly focused on the use of the NanoAssemblr Ignite. So this is where you can optimize the manufacturing parameters. So that's the microfluidic parameters like the total flow rate and the flow rate ratio. And that together with our GenVoy-ILM products, where you can optimize things like your N/P ratio, so an important formulation consideration there, together with your RNA in whatever kind of design form it takes, and provides a platform for you to be able to quickly screen different RNA constructs that you might want to make, different things that you're conceptually making at the RNA stage, and be able to determine and optimize how that would work. And then towards the end of this presentation, I'm going to look a little bit at how you would transition away from using GenVoy-ILM, which is a research use only reagent, onto our proprietary lipid mixtures, which are there for taking through to clinical and commercial applications. So just to reiterate, it's available as a ready-to-use lipid mixture on the NanoAssemblr platform for which it's optimized. It's a similar formulation to the licensed RNA-LNP vaccines. So what I mean by that is in terms of mole percent of the ionizable, the cholesterol, the helper lipid, and the stabilizer, it's very similar in terms of proportionate composition to that of Pfizer-BioNTech and Moderna's vaccine. So really what we wanted to do in this study was kind of show how we could use that to demonstrate how GenVoy-ILM and the Ignite could be used in an early vaccine development workflow. And so an early development workflow might look like this. And this is essentially what we're taking you through. I'm going to show you some of the data in a minute. So in terms of LNP formulation development, we have our Ignite instrument where we can investigate the microfluidic production parameters. So that's what we did. We varied the total flow rate, the flow rate ratio. And then in terms of the formulation, we looked at the N/P ratio. And we did this with mRNA, which I'm going to touch on. And we also did this with self-amplifying RNA, which is another kind of in-house made P&I product. And next year within the BMC, we're going to have a drug substance manufacturing line for offering our self-amplifying RNA vectors as well. So really we wanted to look at the impact of these formulation and process parameters on the physiochemical parameters of the LNPs. But then really from there, we wanted to determine the activity in vitro first in some cell lines, most notably kind of HEC293 cells. And then from there, we wanted to do an in vivo biodistribution study. So looking at how these LNPs are distributed within mice. And then we wanted to do an immunogenicity study, kind of culminating that body of work. So in terms of LNP formulation development, at first, we really looked at optimizing the flow rate ratio with our NanoAssemblr Ignite process. We looked at optimizing the N/P ratio. So that's the ratio of the nitrogen on the ionizable lipid to the ratio of phosphate on our RNA backbone. And we looked at optimizing the total flow rate. And all of this was done with a design specification in mind. So what we wanted to achieve in terms of size, entrapment, PDI. And with that, we were able to land on a set of manufacturing parameters that gave us robust production. And what I want to talk about a little bit on the next couple of slides where I have some data is how we did that with poly-A in the initial instance. So this is a really good kind of strategy for using an RNA surrogate. So RNA itself being quite expensive. So we could use poly-A at that stage and then transition from there onto our mRNAs. So we have firefly luciferase and an EGFP mRNA, and then we transition from there onto our self-amplifying RNAs. So seeing how we could optimize some of these early parameters, at least with regards to their physiochemical parameters, using poly-A, and then transition over those kind of manufacturing steps to mRNA and then self-amplifying RNA. And we did all of this with our GenVoy-ILM reagent, but we also looked at benchmarking this against essentially what is the Onpattro MC3 based composition. So I have here some of the kind of bridging study data that we did. So after those initial studies looking at process parameters for poly-A, we then transitioned over to look at mRNA and self-amplifying RNA. And what we could show is that with the manufacturing process that we'd optimized at that stage, we were able to kind of retain those critical quality attributes as we moved across to the RNA of choice in our vaccine program, which was the self-amplifying RNA. So we had retention of size, which you can see on the graph on the left, across all of these RNA types, roughly similarly. And then we had a PDI of below 0.1 in most cases across all of these formulations. So that's after downstream processing and after sterile filtration. We had consistency in the charge near neutral. And in terms of encapsulation, we did have a little bit of RNA dependency for specific self-amplifying RNAs, but in general, our manufacturing process was fairly robust, giving greater than 80 and in most cases greater than 90% entrapment as we scaled across from poly-A to mRNA to self-amplifying RNA. And so the next thing that we wanted to do from there was take this in vitro. And so, in a kind of standard flow cytometry workflow, essentially we wanted to perturb what the kind of dose response was of the LNPs encapsulating both saRNA and mRNA. And we wanted to look as well at the N-to-P ratio. So although in kind of our initial poly-A studies, we didn't necessarily see too much of a shift in the physiochemical parameters with respect to N-to-P ratio, it's still an intrinsic factor in itself. So changing the N-to-P ratio, although you may not impact something like particle size, you still physically have a different composition with more lipid present in the lipid nanoparticles. So we wanted to investigate that. So taking this kind of standard workflow, I have some histograms here from that work. And what we can essentially see is that, this is where GenVoy-ILM could be used in combination with, say, an mRNA, so one we've obtained off the shelf here, as well as kind of our developmental self-amplifying RNA to check certain parameters. So doing a dose response here, we can see a shift in the mRNA in both transfection efficiency and mean fluorescence intensity. But crucially in our self-amplifying RNA design, we only see a change with respect to the applied dose in transfection efficiency, but we see almost the same mean fluorescence intensity with respect to dose. So that kind of telling us, giving us really good indication that our self-amplifying RNA, the replicon element in the RNA itself is working as intended. So it's a really good kind of checkpoint at this stage before moving forward. And so from there, we really wanted to take this in vivo. I'm going to speak about that in a minute. At this moment, we also wanted to investigate N-to-P ratio, like I mentioned. So, like I said, we did this in poly-A. As you can see, we got kind of across the N-to-P range of 4, 6, and 8, fairly consistent physiochemical parameters in terms of size, PDI, encapsulation, and ZISA potential. And as we transitioned across to mRNA, we got similar physiochemical parameters. But like I mentioned, N-to-P ratio is a factor in itself. We are changing something within the composition, so we wanted to see how this would affect in vitro expression. So first we studied uptake. So this is using GenVoy labeled with a lipophilic dye to study how the particles are physically uptaken. In terms of absolute lipid uptake, so referencing against a calibration curve, we can see obviously as we apply a higher N-to-P ratio, so this is all done at a fixed RNA dose, so we're obviously applying more LNP, you do get more uptake. However, in proportion to the applied dose, we're getting consistency in terms of uptake across each N-to-P ratio. And then the crucial thing is when we do this with our EGFP mRNA, what do we see? And so in the same flow cytometry workflow, we see good viability across all of the N-to-P ratios in terms of transfection efficiency. So the second graph down, we also see approximate consistency in terms of the number of cells that present GFP positive. In terms of mean fluorescence intensity, however, we do see slightly more expression, so slightly more protein expression for N-to-P 6 and 8. So carrying through into our in vivo studies, this is one of the kind of optimization steps that allowed us to select N-to-P of 6, carrying that forward. So if I go into some of our first in vivo data, basically we did an IVIS study where we administered our benchmark LNPs and our GenVoy-ILM LNPs, encapsulating both the self-amplifying RNA and the mRNA for EGFP. And in terms of biodistribution, we administered these intramuscularly. We administered the mRNA at a dose of 5 microgram per leg and the self-amplifying RNA at 1 microgram per leg. And we saw retention at the injection site, as you might imagine, for an intramuscularly administered vaccine here. And then in terms of protein expression, which is what we were mainly interested in, we see differences in the kinetic profile between the mRNA and the self-amplifying RNA. So again, giving us good evidence that our self-amplifying RNA construct, within that, the replication element is working in vivo. And another point to note here is that kind of the self-amplifying RNA is dosed 5-fold lower. Yeah, if we were to plot the area under the curve for each of these, it's approximately similar. So that gives you a good indication that we're, tapping one of the advantages of our self-amplifying RNA, which is that we can afford a lower dose with the same kind of efficacious results, at least in terms of protein expression. And so kind of all of this is coming together towards our first immunogenicity study. So what we wanted to do from there was take the manufacturing process that we'd optimized together with some of those formulation parameters that we screened, and then take our self-amplifying RNA construct, in this case, where it's encoding for an antigen of interest. So we took the SARS-CoV-2 full-length spike protein as an example, and we encapsulated that in on our MC3 benchmark formulation. And then we administered these LNPs at day 0 and at day 28. And we collected serum from the mice at day 27 and then at the termination of the study. And what we saw was good IgG titers in our GenVoy-ILM administered LNPs, both pre-boost or both post first dose, should I say, and then with an increased post second dose. And we also did a little bit of isotype investigation into the IgG2A to IgG1 ratio and found that we have a Th1 based skew in our immune response there, which is good for safety in these kinds of vaccine design and what you would expect for other kind of RNA-LNP vaccines. So at that point, I want to conclude a little bit on GenVoy-ILM and speak then about how you can transition over to our proprietary lipids. So essentially, GenVoy-ILM is available as a ready-to-use lipid mixture. It's optimized for production on the NanoAssemblr platform. It's similar in terms of mole percent to the licensed RNA-LNP vaccines. And what we've done here in this study is really show how you could take that reagent together with the NanoAssemblr Ignite, entrap different RNA types, play around maybe with the composition and some of the design elements of your RNA together with GenVoy to be able to optimize that, and then get to a point where you are kind of happy with the immune responses you're getting with that. And then you can begin to think about transitioning onto the proprietary lipids. So if I speak about that for a minute, essentially beyond our research use only reagents, Precision NanoSystems has a lot of lipid mixtures that are there for other applications and are available for licensing to the clinic and for commercial applications. And so within our lipid library, we have some lipid mixtures which are optimized for different aspects. We have lipid mixtures that are optimized for ex vivo delivery to T cells. We have lipid mixtures that are optimized for delivery for protein replacement therapy, particularly. And then here, obviously, we have LNP mixes which are optimized for vaccine design. So what we could potentially do at this stage is then supply you with lipid mixes to be able to screen for transitioning away from GenVoy-ILM. And so, here's some data from some of the lipids in our proprietary library. So going along the x-axis, these are essentially, it's a lipid composition, swapping out different ionizable lipids and showing that within our proprietary library, we have many ionizable lipids that are kind of optimized for vaccine application. And so you can see in this study as well, we've used the same design that I did for the GenVoy-ILM study, where we've administered LNPs both on day 0 and day 28, and collected sera, particularly at day 42 at the end of the study. And then, this is just showing an example of, again, some of the lipids from within some of the lipid mixtures from within our proprietary library being utilized in a non-human primate study. So essentially what you can do is then, after you've optimized with GenVoy-ILM, maybe conducted your first immunogenicity studies, you can look then at moving away onto other mouse studies for confirming your immune results at that stage, and then scaling up and doing your kind of late preclinical and second animal studies. And so with that said, just to reiterate that within the GenVoy platform, we have GenVoy-ILM as a research use only, ready to use lipid mix, which can be there to kind of accelerate that RNA development program. And then from there, we have GenVoy-ILM within the proprietary lipid library, which can be utilized to transition from that research use only reagent over to something that is available for licensing through into clinical and commercial applications. So with that said, there's a lot of people to acknowledge with regards to the work. I think particularly Natalie Orr, Samuel Clark from PI, and Yvonne Perry at the University of Strathclyde. I'd like to thank them in particular, but there's plenty of other people at Precision NanoSystems and beyond who contributed in some way to this work. So thanks very much for listening. I'd be glad to field any questions.
Thanks very much, Ed, for your extremely interesting talk. So I think I'm going to stop here. Thanks, Ed, for you to join us and share your experience with us. And thanks to everyone for joining us today. And see you at the next Tea Time webinar series. Thanks, everyone.
Great. Thank you.